process. And we haven't addressed everything yet, but we will get to there. Okay. So we have our cell. The cell requires energy. It's going to have an ultimate need to regulate this, not going overboard with the consumption or production of energy in both manners. So there's a very fine balance that it needs to obtain. We know a cell's happy because we see this particular process, the cellular oxidation is decreased. And we're going to talk about oxidative phosphorylation towards the end of this lecture series. The overall concentration of the NADH, which majority of the NADH is going to come from the TCA cycle, which we'll hopefully see next week. The TCA cycle is going to be this hub of producing these electron carriers, and we're going to see those. But the TCA cycle has to be fed from the glycolytic pathway by pyruvate, and that is going to basically be changed into a compound, which we already know, acetyl-CoA. So in a happy cell, we're going to see an increased concentration of NADH overall, which will inhibit the TCA cycle. With inhibition of the TCA cycle, we're going to see then pyruvate be moving through, in some cases, or other sources of material to produce the acetyl-CoA. The acetyl-CoA is not going into the TCA cycle, because that's the interlinkage between the pyruvate, acetyl-CoA, and TCA cycle, to make more NADH. So we've got this very fine balance going through, and it occurs through this enzyme, which we'll study in some depth a little later on, the pyruvate dehydrogenase complex. Now, the pyruvate dehydrogenase complex is this gatekeeper between glycolysis and the TCA cycle. But it's also the gatekeeper to switch on the process of basically gluconeogenesis, because what it's actually doing is prohibiting the consumption of pyruvate to produce more acetyl-CoA, which will feed the TCA cycle to make more NADH. So in this case, we see the pyruvate dehydrogenase complex being halted. We're going to see then the decreased concentration of acetyl-CoA coming from the degradation of pyruvate. And we're going to then start seeing the absolute switch in regards to now an increasing concentration of pyruvate, which is going to actually increase the pyruvate carboxylase activity, the one we showed in the mitochondria, which is then going to go through the process and produce phosphoenol pyruvate, stimulating gluconeogenesis and provide glucose. So from this little picture, we can see the important play of substrates, such as NADH, acetyl-CoA, the issue of pyruvate playing a role with the pyruvate dehydrogenase complex, and also then the pyruvate concentrations changing at the expense of the acetyl-CoA, because we're not feeding pyruvate into the TCA cycle anymore through the pyruvate dehydrogenase complex, and therefore we'll see additional decrease in production of acetyl-CoA, but we've still got a lot in there because we've basically stopped. And we also have other sources of acetyl-CoA, which we'll see a little later on. So in this case, it's just an illustration of the complexity. And remember, all these require transport systems. They require potential diffusion kinetics. So we also might have enzyme, particularly with this guy here, the pyruvate dehydrogenase complex is actually a complex protein of a number of subunits. And it basically hands the compound around, the substrate around to do its work. And again, basically, we're going to be inhibiting that particular process because we're in a happy cell complex, because that acetylcholine, as well as the NADH, has some level of sensitivity on this key enzyme, the pyruvate dehydrogenase. So what I'm trying to illustrate here is this connectability, and we're going to have a little bit more talking about the connections that we have studied already. And you'll be amazed in regards to what you know and how we're now going to connect the individual units, just to think about the process of glycolysis, gluconeogenesis, and the results of those particular actions in regards to the biochemistry. So we have a key compound which is called fructose 2,6-bisphosphate. Now, the 2,6-bisphosphate is separate from the 1,6-bisphosphate because there is a different phosphorylation in the position number 2 instead of the position number 1. So the fructose 2,6-bisphosphate is a key hormonal, it's a product from a hormonal regulational seesaw in regards to switching from glycolysis to gluconeogenesis. And this is our key point in regards to the movement of the fructose 6-phosphate, committing it to gluconeogenesis, or committing the fructose 1,6-bisphosphate to glycolysis. It is this sole enzyme, this sole substrate, in its concentration presence, acts as a allosteric modulator, having an effect in regards to one enzyme. We'll talk about the fructose bisphosphatase number 1 in a minute, but we also have our old friend, the phosphofructokinase number 1, and this is a positive allosteric modulator on this one and a negative allosteric modulator on the other. So it gives us this capacity to switch between one system and the other by using an intermediate, a byproduct. Remember, we have fructose 1,6-bisphosphate, so it doesn't take too much of an enzyme to actually shift that additional phosphate position in number 1 to the position number 2 to produce this guy. So there's going to be an enzyme that does that. We'll have a look at that in a second. So we have the fructose 2,6-bisphosphate, which is not an intermediate, but is a compound that is derived from the intermediates of the glycolytic pathway in regards to the reflection of the concentration of another hormone. Now, this is a peptide hormone, very similar to insulin, but has the opposite activity. This is glucagon. Now, glucagon concentrations in the blood are going to reflect importantly when blood glucose rises. So in this case, falls. Insulin reflects increased blood glucose levels. Glucagon reflects decreased blood glucose levels. Because what we saw with insulin's activating the transporters to move the glucose into the cell, where the glucagon is trying to maintain the circulatory level of glucose by then causing the glucose to come out of the cell. But if you think, where's that glucose coming from? It's coming from our stores in regards to our glycogen, which we'll have another look at again, and also the issue of now going through gluconeogenesis. So there's going to be some promotion. There's, again, a seesaw between insulin and glucagon to give you this process of very fine-tuning regulation. So the fructose 2,6-bisphosphatase activates the phosphokinase number 1, which will then promote, in this case, if this compound is abundant in regards to a positive steric modulation, we're going to see the consumption of the fructose 6-phosphate to give it that dedication to fructose 1,6-bisphosphate, which commits it to the process of glycolysis. If the fructose 2,6-bisphosphate is also present, we're going to see an interactivation of inhibiting the negative process. So in this case, with the fructose 2,1,6-bisphosphate, if the 2,6-bisphosphate concentration is depleted, it is going to basically then have a positive effect on this guy here. So if we decrease the concentration here, we're going to see the fructose 1,6-bisphosphate not going into glycolysis, but being moved back to fructose 6-phosphate to go into gluconeogenesis to produce glucose. And that is because of its inhibitory action on the fructose bisphosphatase enzyme number 1 when it is present. If it's depleted, it's not going to have that allosteric modulation, and therefore the enzyme will actually kick in. So we have the fructose 2,6-bisphosphate activates the phosphofructokinase number 1, but in unison at the same time inhibits the fructose 2,6-bisphosphatase. So we've got this seesaw balance happening. So again, when the concentrations of the fructose 6-phosphate increase, the concentrations of the fructose 2,6-bisphosphate also increases because it has effect on this particular enzyme. Okay, so again, we're going to have this particular system as a key regulator. 2 steps of choosing gluconeogenesis over glycolysis. Also note, only one of the systems, and we know this one, the phosphofructokinase number 1, is consuming ATP. The other one is hydrolyzing the phosphate group. So this one is placing a phosphate group on, obviously, to get the fructose 1,6-bisphosphate. This one is hydrolyzing the number 1 phosphate position in carbon number 1 position on the fructose to give us the fructose 6-phosphate. So again, this kind of very closely guarded regulatory system in making that pointant switch between glycolysis and gluconeogenesis. So it's this regulatory component, and we also have other regulators that come in and regulate these enzymes too. Now, I've stated here, the fructose 2,6-bisphosphatase is actually inhibited by AMP. Now, AMP is indicating to us, let's go back here, AMP is indicating that we need to basically go through consuming AMP, consuming ADP to make ATP. So we're going to ideally promote the process of glycolysis and allow us basically to make more energy. So again, the counterpart though, the glycolytic counterpart, the phosphofructokinase number 1, is going to be stimulated by that same indicator, meaning we've consumed ATP, we've made ADP and AMP, and they're going to be indicators to say, hey, we need now to make ATP because we have an abundance of AMP and ADP, and we're going to switch the enzyme on that's going to do that, which is the phosphofructokinase number 1. But we also have a counterpart also to this mechanism. It's getting a little more complex. We're having multiple allosteric modulators, both for the positive and the negative effect, because the phosphofructokinase number 1 is also stimulated by a distant pathway within the TCA cycle, and that is by a compound called citrate, and we'll have a look at that in next week. So citrate has the capacity in this case to bind to the phosphofructokinase number 1 and ultimately slow down or inhibit the pathway that leads to its production. Because at the moment, there's enough of that citrate turning around in the TCA cycle to make the NADH, the FADH, and the GTP, which we can make ATP out of. So also, if the cell is happy and we've got abundance of ATP because we basically prohibited the fructose 2,6-bisphosphatase, we promoted the phosphofructokinase number 1 to do its job, and now we're seeing an increase of ATP being present, we're going to basically switch off that particular source to make sure we are not oversaturating the system with ATP. And we're going to do that by inhibiting the phosphofructokinase number 1 enzyme. So let's have a look at these seesaws where we have high concentrations of acetyl-CoA and citrate are going to weight down the system to then basically lighten the process of gluconeogenesis, because these guys are indicating that the cell is producing a lot of NADH, which is going to be ultimately the electron carriers, as well as FADH2 to the oxidative phosphorylation system, which will make us ATP. So we've got enough stuff in reserve coming through. But as this depletes, we're going to change our system. In this case, we're going to be sensitive to the breakdown of ATP to AMP, specifically via ADP. So when those concentrations of AMP start increasing and become heavy, we're going to be light in glycolysis and be heavy in gluconeogenesis, because what we're doing is we're increasing that phosphofructokinase number 1 activity to give us the capacity to make more material, to produce more ATP. And also, we're going to be mobilizing the source for production of ATP in the glycolytic pathway by increasing the glycogen phosphorylase. Okay? So there's a very rapid and reversible allosteric modulation mechanism at multiple points that are coming from within inside the cytosol of the cell to allow us this regulational situation to occur. But also, we have external stimulation. And we already know this because we understand the issue of insulin and the insulin receptor and the insulin binding substrate receptor number 1. And now this compound, this peptide called glucagon. Now, glucagon also stimulates in regards to the epinephrine receptor too. I mean, the epinephrine receptor is a separate GCPR, but also has a similar impact in regards to glucagon, because obviously the fight and flight mechanism, if we've got that GCPR activating, increasing the calcium via the IP3 receptor and the endoplasmic reticulum, releasing that, activating the phosphokinase B, doing those phosphorylational events. Because here, with insulin, glucagon, and epinephrine, it's this issue of phosphorylation and dephosphorylation that happens in regards to the process of regulation. This happens inside the cell, but the communication from outside the cell by the hormones takes a significant amount of time in regards to having localized materials, such as the fructose 2,6 bisphosphate moiety, which is already in the system, already in the cell, already localized, having the capacity to have a similar effect in regards to the regulation of glycolysis and gluconeogenesis. So as I'm saying here, the phosphorylation and dephosphorylation events, they could have both positive and negative effects depending on what type of system you're looking at, but they take time to occur, where we have localized allosteric modulators derived from intermediates in regards to allowing us to have a allosteric modulation at the localized site, which is going to give us a more rapid response. So again, we've got to keep thinking of all these types of responses, the slow, the fast responses, the sequestration, meaning the compartmentalization, the kinetics of the reaction, the lipid rafts that bring all these particular proteins together to inhibit or decrease the reliance of diffusing molecules within the cytosol. Again, localization, compartmentalization, concentration becomes a very important role. So when we see in ourselves, and some of you are mostly experiencing this now, if you've not eaten your breakfast, that you have a decreased blood glucose level. This is going to trigger the secretion of the hormone glucagon, which is going to go to the liver, and the liver is going to say, hey, I've got this hormone, it can't go through the lipid membrane, I'm going to use a GCPR, a receptor, to actually trigger a cascade of information through the lipid membrane and allow phosphorylation events to occur, which will cause the issue of production of more glucose by the stimulation of an enzyme system. We knew if we've got glycogen, we're going to be using that store, or if the glycogen store is finished up, we're going to be producing more glucose from the process of gluconeogenesis. But the important thing is the liver, and I think I've mentioned this before, will actually sacrifice its own requirements of glucose to supply and support the body. So in this case, we're going to see some very interesting biochemistry occur in regards to the liver hepatic cells themselves. They will sacrifice themselves in regards to maintaining a glucose level for the rest of the body. Remember I said, your brain here is basically a glucose sink, and your body is basically here to basically maintain that glucose pool to keep this guy going. Without glucose in the brain or going to the brain, you're dead. Simple as that. So again, I'm going to emphasize, the fructose 2,6-bisphosphate, an allosteric modulator, it's going to promote the protein kinase, the phosphofructokinase number 1, at the expense of the fructose bisphosphatase. It's basically going to see then the decrease of the fructose 2,6-bisphosphate, which is then going to activate then the fructose bisphosphatase to promote gluconeogenesis in the liver. Now, so we have this little seesaw, and it's best to actually see it in this particular seesaw. The fructose 2,6-bisphosphatase is that key point, that pivotal point. Depending on which way it's going to go, it's going to basically change the system of glycolysis to gluconeogenesis. In glycolysis, we're going to see an increase of glucose coming through the glycolytic pathway. We're going to see an activation of the phosphofructokinase number 1 at the expense of the fructose bisphosphatase to satisfy that. And again, once we shift this over, we're going to then see a change in regards to the decreased concentration activating and reversing this particular process to promote, importantly, then gluconeogenesis. So the liver is very, very important in doing this. And we're going to see the opposite with the correct increase in concentration of the fructose 2-bisphosphate, activation of the phosphofructokinase number 1, which will then give us that glycolytic pathway at the expense of gluconeogenesis in the liver. So again, this single pathway having this seesaw dependent on the fructose 2,6-bisphosphatase, the fructose 2,6-bisphosphate is activation on the fructose bisphosphatase number 1, and the activation also of the phosphofructokinase number 1. So again, it's a little complex, but if you could imagine these seesaws, you're going to have a pretty good understanding. So remember, glucagon is a hormone, a peptide. It's going to work through a GCPR to promote the production of more glucose. Now, it's going to have systemic action, but most of the action we are concerned is actually going to focus on the liver. So this gives us a little picture of what's occurring overall. We have the bloodstream in regards to trying to maintain that particular concentration of glucose. The liver cells are going to do their work by metabolizing glycogen and producing glucose 6-phosphate to give you more glucose through the dephosphorylation, which will then be transported, as we saw with the transport system T3, T2, and T1, allowing us to supplement that glucose pool into the bloodstream. That can then go and be utilized by the cells in the muscle particular, which will then utilize the glucose, make glucose 6-phosphate, pyruvate, to then go full metabolism, making lots of NADH, FADH2, which will ultimately produce tons of ATP, but only if there is oxygen available. If oxygen is not available, we're going to have lactic acid production. That lactic acid is going to start building up and it's going to be shunted off into the circulatory system. In that case, the lactic acid is going to be metabolized by the liver by going through with the consumption of moving those NAD plus to make NADH to give us pyruvate, which we can then also metabolize through the TCA cycle, or we can take that pyruvate and produce some other molecules, which we'll have a little look at later on. Now, the liver also plays a real important role in regards to circulating surplus glucose. So if there's a surplus amount of glucose, because you've just eaten a very large sugary meal, that glucose will come in, produce glucose 6-phosphate. It'll actually go to the pyruvate system to produce carbon dioxide and water at the expense of degrading the pyruvate to produce NADH, FADH2, and GTP by the TCA cycle. Pyruvate will also be utilized in regards to producing some biological precursors, which we'll talk about later on. But ultimately, if we have too much energy, and now we've got a surplus of glucose 6-phosphate, we're going to produce glycogen. And we're going to store that glucose in the liver. But also for the same situation, we're going to see an increased glucose level in the bloodstream. We're also going to see this muscle and even the cardiac muscle respond to that glucose concentration, where we'll see glucose coming through, again, glucose 6-phosphate, and producing glycogen as a store. But this is an intermediate store, a small amount of storage material to actually allow us to do the normal process, depending on the depletion of glucose from the bloodstream. We also see the same in the cardiac muscle in regards to the issue of utilizing that glucose 6-phosphate as a source to provide a store of glycogen. But again, a small storage. Most of the storage of glycogen is coming from the liver. But as you now have seen, most probably, number 8 are amino acids. Now we're going to study this most probably next week in regards to the role of our amino acids now coming in. Remember, we talked about glucogenic amino acids. Those amino acids are now going to come in and play a role in both providing pyruvate or transforming to produce pyruvate, as well as other intermediates, as well as feeding the gluconeogenic pathway if we need to make glucose. So those glucogenic amino acids are going to feed into the TCA cycle and provide us the capacity to produce our friend oxaloacetate. So keep that in mind. Oxaloacetate is going to be a very key component in the process of glucose formation from amino acids. So we'll talk about that a little later on. So we're starting to make this picture and connection. So we know that amino acids are going to be playing a role in glycogen, in glucose synthesis. We're going to think about glycogen as a store, when it needs to be depleted, when it needs to be topped up. And we also know, as you can see here, with this simple picture of compartmentalization, there's going to be some different enzymes. We've already talked about the hexakinases in this particular step, which have some different capacities in regards to allosteric modulation, as well as their kinetic process. So again, not all enzymes are the same. Not all hexakinases are under the same jurisdiction. They do a job as a hexakinase would, but they have different modes of regulation and allosteric modulation. So again, dependent on the cell type, there's going to be some differentiation. So this is a great little picture in regards to kind of putting things together. And we're going to explore this a little more in the next lecture. So I want you to understand the glycogen and glycogen synthesis, the regulation and allosteric modulation, sequestration in regards to the compartmentalization, compartmentalization to also allow you to have regulation. We should understand what hypoxia does in regards to the cellular system, where we're going to be pushing things to produce lactic acid. We now know there's a difference between some of these enzymes in our main metabolic pathways, depending on what they're actually doing in regards to the cellular type, such as the hexakinases versus the liver versus the muscle. We should understand about the happy cell in regards to some of the components and how we keep that cell happy in regards to meeting the energy requirements. We should also know, and we do know about insulin, but we've now introduced a new hormone, glucagon, in regards to doing the opposite to what we see with insulin. So again, I think we need to have a look at these in regards to the increased blood glucose levels and the decreased blood glucose levels. The issue of epinephrine, we already know that in regards to the fight or flight system, and insulin with the insulin receptor substrate number 1 stimulation and the transport stimulation, and glucagon doing a very similar effect with a GCPR. We think about the regulation of gluconeogenesis and obviously the coordination to ensure efficiency between that switching with the fructose 2,6 bisphosphate and its impact on the phosphofructokinase number 1 and the fructose bisphosphatase. And again, also this central role of the phosphoglucomutase when we're looking at the breakdown of glycogen. And we have to understand, importantly, the production and degradation of glycogen as an energy source through that dephosphorylation, as well as the linear versus the branching chain in regards to the 1,4 versus the 1,6 degradation of glycogen. So again, these are all important things that are occurring in your body now. And Without it, you'd be dead. Finishes that lecture. So let's get on to lecture 27. Okay. So you're all still with me? Is it getting a little more exciting? Kinda, yeah. What's he talking about? I have no idea. Some of this will be on exam number 3. So just keep aware of that.